Good evening to all. Today I uploaded that video, but the audio was not clear. Okay, we were discussing translation. The last class we studied genetic code and tRNA structure, and I uploaded one video. Did you see that video? In detail it is described. This part is translation, and this translation it mainly involves three processes. That is initiation, elongation and termination. Initiation, elongation and termination, and this process occurs in the ribosome. This translation process occurs in the ribosome. This ribosome it consists of two subunits, that is smaller subunit and larger subunit. Smaller subunit and larger subunits. And when this smaller subunit encounters with this mRNA, then the initiation process starts. This translation process starts when this mRNA encounters with this smaller subunit. After that, this larger subunit combines with this smaller subunit, and this mRNA it consists of different nitrogen bases. Different sequences are present. It runs from 5' to 3'. It runs from 5' to 3'. And then it reads from 5' to 3'. And this initiation process it starts from when this initiation codon AUG, when this initiation codon is recognized by tRNA, the corresponding complementary tRNA. When this initiation codon AUG it is recognized by the tRNA, that is complementary tRNA, and that time this initiation process starts. That process it is known as initiation. The initiation process starts when this start codon, initiation codon is recognized by the tRNA, corresponding tRNA. Then this translation process proceeds, and the corresponding amino acids are added to this one. These amino acids are joined together by peptide bond, and this peptide bond it is formed for the formation of peptide bond. It needs some energy, that is by the process of known as amino acylation. It is by the process of amino acylation. This one for peptide bond formation it gets energy by amino acylation process, and this translation process proceeds, and this last when the initiation when it codes this one in the elongation process proceeds, it reaches to the termination codon. The termination codon it is UAA, or it may be UAG or UGA. The stop codon it may be UAA, UAG or UGA. These are the stop codons. When it reaches the stop codon, it is not coding for any protein, but it is when it reaches there, some releasing factors are produced, and it leads to the termination of this process, and it helps to release the polypeptide chain from ribosome. Like that, the termination process occurs. When the translation proceeds, when it reaches to the stop codon, some releasing factors are produced, and these releasing factors it helps to terminate the process, and this polypeptide chain is released from the ribosome, and like that this translation process occurs. In translation, three processes are there involved, that is initiation, elongation and termination. It helps to produce a protein. Translation refers to the process of polymerization of amino acids to form a polypeptide chain. Translation refers to the process of polymerization of amino acids to form a polypeptide chain. And here you can see this 5' and 3' end after the initiation codon, before the initiation codon and after the this stop codon, you can see some codons are present, some sequences are present, and such regions are known as untranslated regions. That regions are known as untranslated regions, that are very essential for translation, that are very essential for efficient translation. Untranslated regions. Then in between the translational unit in mRNA, it is in between the initiation codon and stop codon. this one, this translation unit that is in between the initiation codon and stop codon. This is the translation process. Involves initiation, elongation and termination. It occurs in the ribosome. Ribosome consists of two subunits, smaller subunit and larger subunit. And in the smaller subunit, when mRNA encounters this translation process starts. That time this larger subunit combines into that one, and this larger subunit consists of two regions. One region for peptide bond formation, one for the entry of the next tRNA. There are two regions in the larger subunit. Okay, thank you.